Another day and another IgG4 publication. This is now the second one of this year, second one in January already. This one comes from Puerto Rico. This is also now a third publication that looks at Fc gamma re effector functions. I'm going to explain that in detail, what that means. This is one of the more important aspects to understand in terms of with regards to these IgG4 antibodies and their potential clinical role. So we're going to get into detail of that. And it's a second now paper that is comparing Moderna effect versus Pfizer effect of these mRNA vaccinations. Because remember, it appears that the vast majority of people who take mRNA shots, about 98%, end up producing these mysterious IgG4 antibodies. All right, with few exceptions. And check out past videos to learn more about what would be the circumstances with regards to when the mRNA vaccinated would not be producing IgG4 shots. And again, it's still an incomplete story. In total, this is now a 13th publication, 1-3, with regards to showing that IgG4 antibodies increase dramatically especially after the third shot. These individuals, what they were looking, but this is really interesting. Another very, very valuable aspect of this paper is that they were comparing individuals who had 3 mRNA shots, and they looked at 10 people who had Moderna shot versus 10 people who had 10 people who had 3 Moderna shots versus 10 people who had Pfizer shots, 3 Pfizer shots, and either one of those groups were followed up with Novavax vaccine booster. So Novavax, remember, is a protein vaccine. So you get injected with a certain amount of spike protein only. And they juxtaposed that with people who had 4 Novavax shots. So, and they were measured about 18 of those individuals. So what did they see? So basically, and my name is Dr. Mikola Rashik of Merigenomics. So let's get started. So what did they see? First, we're going to talk about IgG4s, and basically they mentioned that Novavax protein vaccine, they measured it between only 3rd and 4th shots. So they did not look at before other shots. The also authors did not provide really much information in terms of time between vaccinations, and which is unfortunate. These are actually very valuable informations right now while we're trying to work out what is going on with regards to these IgG4s. So that's important knowledge, but nevertheless, they looked at these subclasses of IgG1, 2, 3, 4 between 3rd and 4th shot. And basically, Novavax, while it did show increase, almost 2-fold increase between 3rd and 4th Novavax protein vaccine shot, that was actually negligible or very small in comparison to what was observed for those who were mRNA vaccinated first and then had Novavax booster, while the total amount increase in IgG4 was also not that big between 3rd and 4th shot in those individuals. It was between 2 to 3-fold increase. But the major increase, the major difference was that after 3rd mRNA shot, these individuals were already presenting with very high levels of IgG4s, like what we've seen all along in other literature I've discussed. And basically, it was about 75 times higher, the amount of IgG4 in the mRNA vaccinated individuals who had Novavax booster than individuals who just had Novavax shots, 4 Novavax shots. So huge difference. This is why the authors of that paper concluded that Novavax shots do not really induce IgG4s. Okay, so that's basically the big part with regards to IgG4 levels between those individuals. But now let's continue. And they also, another really valuable part of this paper, as I mentioned, is the fact that these individuals, the authors of this paper, looked at these Fc gamma effector functions. So what am I talking about, right? Let's break this down. This is really important, because really what we're talking about is this. When you have antibodies, antibodies, of course, are supposed to bind, attach themselves to the pathogen of interest, right? This is why we're vaccinating, so that we can produce antibodies that will attack the virus once we get infected and clear the infection. That's the ideal part. But these antibodies also have these stalks sticking out. And these stalks are called Fc gamma. And they can be recognized by immune cells. And this is partially how viral infections are cleared. It's not just that you neutralize the virus by binding antibodies to it so that the virus no longer can infect cells because it's being blocked from achieving that. When you bind these stalks, Fc gamma stalks of these antibodies by other immune cells, these immune cells can do a couple things. They can either swallow up the complex and then it would be destroyed. That's called antibody-dependent cellular phagocytosis. I know it's a mouthful. Or they can destroy cells that are already infected and present the, say, spike protein on the surface. That's referred to as antibody-dependent cellular cytotoxicity. They measure both of these. They also measure the effect of complement system. Complement system is basically, think of chain reaction of a bunch of molecules attaching themselves together to a pathogen, and that can also trigger certain immune cells to recognize that and destroy cells that are infected. So, and that's referred to as antibody-dependent complement cytotoxicity, okay? And these authors measured all three of them. They did not measure it directly. They looked for the capacity of this binding between the receptor of interest that's supposed to recognize these stalks, and they measured this indirectly. I'm not gonna get into the details of how they did it because it's too complicated, but I can tell you that basically it's like chain reaction of Lego pieces where you attach one thing on top of another and then on top of another, and then you can see something like light emission or chemical reaction, and then this is how you measure that something is happening. It's really ingenious what they did, but the point is that they measured these effector functions. So, and what they showed is the following, that after 4 Novavax shots, these effector functions between third and fourth shot, they increased in their amount, which is great news. We want to see that. It clearly means that these effector functions, why they're valuable, is because not only means that antibodies are neutralizing the virus, it also means that it helps with the viral infection, because it can remove, it would indicate that cells that are infected could be removed, and that's how it's well known that that's how you can clear the infection. These effector functions are very important in helping us deal with infection. So that's what they observed. Now, they did observe increase in these effector functions between third mRNA shot and the fourth Novavax booster. It just was not as much as what was seen with Novavax alone. But nevertheless, that's really potentially good news. I can tell you that those effector functions were definitely much lower in after the third mRNA shot in comparison after the third Novavax shot, which basically means that these effector functions are suppressed and in those individuals, and that suppression might be due to IgG4. Why is that? Because this is where potentially is the problem with IgG4s. Why? Because the stock of IgG4 is made differently than the stocks of the other type of antibodies, in that the stock of the IgG4 antibodies is not recognized by these receptors that are supposed to recognize Fc gamma stock of the antibody. They just don't recognize it as well, and as a consequence, that's one of the problems with IgG4s. They can cloak whatever they're recognized, they can cloak the spike protein, but the spike protein will not be removed through these effector functions. Meaning, say, if a cell has a spike protein on its surface, it reduces the likelihood that these cells will be destroyed. And that's the reason why we need to pay attention to these, and why if these are effector functions are affected to a great enough degree, why this might actually result in certain clinical complications. And this is why we need to study this a lot more. Unfortunately, as I mentioned, this is only a third paper that has investigated this, but hopefully we'll get more data as we study this further. So why is this good news? Is that while clearly they are showing that these effector functions in mRNA vaccinated might be compromised, that they're reduced in quantity, it might be very good news in terms of the fact that Novavax shot increased it. But maybe it is restoring, but there's a trick to this. What am I talking about this? What am I talking about here? So this effector functions can actually be a double-edged sword. And that depends on whether the antibody is a good quality antibody, strong binding antibody, or a weak binding antibody. So it's, as I mentioned, it's been well known. It's been observed before with SARS-CoV-1, the first one, that these effector functions, when they play the role, it can help clear the infection. On the other hand, it's been shown as well that it's irrespective what the antibody can bind, actually, it could influence in clearing the infection. Why is this? Is because of these effector functions, right? So that's good news, seemingly good news. And also it's been shown that if you remove these effector functions completely, and those were shown in mice, you will prevent proper viral clearance. So they are important. But how is this a double-edged sword? In that sometimes if these stocks of these antibodies, these Fc gamma stocks, are grabbed by immune cells, but the antibody that the immune cell was grabbing is a very weak binding antibody, it can cause what is referred to as antibody-dependent enhancement, meaning enhancement of the actual disease. What do I mean by this? Basically, the immune cells that grab the stock of the antibody swallowed up the pathogen complex with the antibody, and then because the antibody is weak, it can release the virus, and instead now you have a cell that got infected. So hence the enhancement of the disease. This has been observed in the past with certain viral infections. And that's where the potential problem is. So when we are seeing this increase in these effector functions after 3 mRNA shots, and if the person took a 4th Novavax protein booster, we then still need to find out, well, is this the good effector function or is it bad effector function? Because depending on whether we're dealing with boosting good quality antibodies or poor quality antibodies. And why do I mention this is because, recall in one of the past IgG4 related videos I made, I discussed the fact that these IgG4 antibodies might be weaker binding antibodies, so not binding as good as a really high quality antibodies, because they are now binding different regions of the spike protein due to the concept called immune refocusing. So what basically happens is that as we keep vaccinating, we might be stimulating the immune system to start shifting what kind of epitopes, or basically targets, these antibodies recognize on the spike protein. And if they are shifting, they might be shifting towards poorer quality targets. And if that's the case, then we still have to figure out whether this restoration of these effector functions with the Novavax booster post mRNA shots is a good thing, or could it potentially lead to antibody-dependent enhancement of disease? So we still don't know. So potentially good news, but we need more information to confirm whether this would be maybe a good way to make sure that these effector functions are not compromised to a great degree by the IgG4 antibodies. All right, that's all I have for you today. Please share the video, Please leave a comment as well. This is how we grow. YouTube likes it, and so do we. And also, last thing, please check out our Patreon account. We post other videos that are potentially more controversial in topic. And of course, check out other past videos related to the IgG4 antibodies that we are seeing in mRNA vaccinated individuals. This is really important for us yet to figure out what is going on here clinically. And hopefully, as time goes by, more and more research comes out. Clearly, this is captivating people because clearly more papers are coming out more rapidly on this topic. So this is definitely something that is now capturing scientists' interest from all over the world. I assume I might have mentioned this one. This particular study was from Puerto Rico. And yeah, hopefully in total, this will coalesce into our understanding, better understanding what are the clinical implications of this massive increase of these mysterious IgG4 antibodies post-mRNA vaccination. All right, that's all I have for you, and see you in the next installment. Bye, everyone.